Hello, hey, I'm Mr. Doc, and this is another of the My eBiology Teacher podcasts. Today we are wrapping up our unit on cellular reproduction by talking about meiosis. Now, we've talked about mitosis already, so we're going to take a couple of minutes to go over meiosis. We're going to talk about how meiosis and mitosis are similar, yet different. And we're going to talk about its importance. So let's go ahead and get started by understanding the concept of homologous chromosomes. Now, it's a big, long, compound word, which basically means two copies of a chromosome that contains the information for the same traits. In other words, the homologous chromosomes, one will have the traits for hair color and eye color and all that other stuff. So each chromosome has a list of the same traits. One half of a homologous chromosome is called an autosome. And during sexual reproduction, we get one autosome from mom and one autosome from dad. So we end up with two or a pair of homologous chromosomes. So we get one half infant. So that means that we're made up genetically of one half from mom and one half from dad. Now, this means that the chromosome from mom has all those traits on it, and the chromosome from dad has all those traits on it. When they pair up, that's when we get into genetics and determining which one's dominant, which one's recessive, and all that stuff, which we will cover down the road. Alrighty. The phases of meiosis are as follows. And by the way, I'm jumping right into this because we've kind of covered mitosis. And if you understand mitosis, meiosis actually is pretty easy to understand. Instead of starting with prophase, metaphase, anaphase, telophase, it's going to go in the same order, except now we're going to add Roman numerals, and you'll find out why in just a second. The first phase of meiosis is prophase 1, and in this phase, we're going to take the chromosomes and they're going to pair up into what's called tetrads. That means that they are going to pair up with their homologous chromosome and they are going to hang out together. However, during this process, crossing over can occur. So basically what happens is these two get together, perhaps a little piece from this one will cross over, and then we have some information from this side and that side, and what that does is creates genetic variations, different combinations of traits that didn't exist before. This is called crossing over, and it's important. It's one of the main reasons why, as a result of meiosis, no two people are identical to each other. So in prophase, the homologous chromosomes pair up in tetrads. During a process called synapsis, crossing over occurs in which some part of some chromosomes will cross over to another chromosome. Now, in metaphase 1, and notice that we have 1, 1, 1, so we're talking metaphase, anaphase, telophase 1, all that. They're not going to line up across the equator of the cell like in mitosis. What's going to happen is they're going to line up by twosies, right? They're going to line up side by side. They're going to do this randomly, which means that we're not sure which is going to go to which cell. Okay, so we could say, and if you'll notice in the diagram up here, instead of all the blues lining up on this side and all the greens lining up on that side, we can have a little randomization, again, creating more genetic variations. And if you also look, we can tell that crossing over has happened on these because this blue one here has that little green spot right there, and in the same spot on the green one, there's a little blue spot, right? So that's crossing over, and then we have this whole randomization part called independent assortment. An independent assortment happens in anaphase 1 where the chromosomes start to split apart. Now, this is different than mitosis because in mitosis, the chromosomes split at the centromere. That's not going to happen here. Because we've lined up like this, a whole chromosome is going to go up and a whole chromosome is going to go down. That's in anaphase 1. Now, in telophase 1, it's very similar. The cells are going to come back down on itself, and we're going to reform, and everything's going to be nice and dandy. Cytokinesis is going to happen. We're going to end up with two cells. Now, here, mitosis would go directly into interphase, and the DNA would copy itself, and blah, 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 blah. That's the difference between meiosis and mitosis. Meiosis does not go into interphase. We're going to go into another round of cellular division, which is going to give us the opportunity to divide again without copying the DNA. All right? Now, the meiotic process is going to continue, and it's going to go through the same phases a second time. So we're going to go through prophase 2, metaphase 2, anaphase 2, and telophase 2, right? In this case, it looks a lot more like mitosis. In other words, the membrane is going to dissolve, they're going to line up in the center, they are going to split at the centromere, and the chromatids are going to separate, and then we end up with two cells. One difference is that these cells are not genetically identical as opposed to mitosis. Another thing is they are haploid, meaning they have half the DNA that we started with. Why? Because we didn't reproduce our DNA. Now this is a good thing because the resulting cells have half the chromosomes. When they add to another haploid cell, sperm and an egg, that cell is going to be diploid, which is exactly what you want. You see, this whole process is called gamete production. Meiosis and gamete production are synonymous. They basically mean the same thing. Meiosis is important because of gamete production. And gamete's a fancy word for saying sperm or egg. Gametes are sexually reproductive cells. Sperm is the male gamete, egg is the female gamete. If gametes were produced by mitosis, then what would happen is they would both be diploid, which means that naturally they would have 46 chromosomes in humans. Well, that means if we have 46 plus 46, we end up with 92. That's not good. And if someone who has 92 reproduces with someone who's got 92, then we end up with 184. And then again, it'd be 368. Right? I mean, what would happen is the DNA would keep duplicating and duplicating and duplicating and duplicating until what would end up happening is we're nothing but DNA. There's another problem here. There's a law of non-contradiction, and it says that we cannot be A and not A at the same time. In other words, I can't be here and not be here all at the same time. Well, imagine if we had double copies of our chromosomes. Well, that would mean that I could be blonde and brunette at the same time. Well, that can't happen. I can't be both blonde and brunette at the same time. I can't have brown eyes and blue eyes at the same time, meaning both eyes, okay? So we have to cut that in half, okay? We have to cut the DNA in half. So that means that in the humans, our natural cell condition, our skin cells and whatnot, they have 46 chromosomes, but our gametes, sperm and egg, have 23, because when 23 meets 23, boom, 46, and that's what it's supposed to be. All right, we got a little tie right there, but that's okay. Now, I want to show you a little review. This one's kind of funny. I got it from unitedstreaming.com, which is a service that North Central uses. It's kind of a fun way of watching meiosis. It's called the Meiosis Square Dance. Let's take a look, shall we?
Stina, hurry! We have to call up soon.
Okay, I'm duplicating as fast as I can.
Ah! There's the music. Quick, call up!
Call it tight. We're teaching my pieces right. And the big one is almost done. Monkey style, perfect one. The picture part is you get down. You'll see the rest over now. How many left with your left hand? Promenade around the land.
Uh-oh, they're crossing over. Ooh, looks like they're randomly assorting. How cool. How'd that happen? So we got some yellow over there, we got some green over here. That looks independently assorted to me. Oh, watch out for your spindle. Now this looks like mitosis, but it's done. It's meiosis 2.
the system we vertically out from once the tree inside of bladder will divide again it's for the equal side. And if we zoom out, you will find living more the second. If you're wondering what's the use, also you get reproduce.
So you can All right, so there you go. I know that was a little over the top, but hopefully you get it. Hopefully you understand that meiosis is a process that happens twice over, much like mitosis only happens once over. Now, there is something that we mentioned in the previous slide, and that is that sperm and egg are produced in different ways. That is to say that meiosis still happens, but the final result is a little bit different. Spermatogenesis is the way that sperm is produced. We start out with a germ cell, which is here, and we end up with 4 viable sperm, each unique. It's not an exact copy of the germ cell, which is why one sperm would result in one type of offspring and another sperm would result in another. In oogenesis, which is egg production, a diploid germ cell gives rise to 3 polar bodies and 1 haploid cell. The haploid egg cell is the only viable, meaning the only cell possible to reproduce offspring. And then we have 3 polar bodies. Those are bodies that will never become offspring and will eventually be destroyed as part of the reproductive process. So spermatogenesis results in 4 equally viable but different sperm, and oogenesis ends up with 1 viable egg and 3 polar bodies, the polar bodies not being part of the reproductive process really because they're going to be destroyed. So let's go ahead and kind of review. First off, mitosis and meiosis are similar in that they are both forms of cellular division. They use roughly the same phases, prophase, metaphase, anaphase, and telophase. The main purpose of both is to create genetic ways of passing on material. In other words, containing genetic material passing on from a parent to an offspring or from a germ cell to a daughter cell. And both processes are asexual in nature. Now this can be confusing because we think of meiosis as being regarding reproductive cells, in other words, regarding sex cells. However, the process of meiosis itself is asexual. The final product will participate in sexual reproduction, but the process of meiosis is itself asexual. Now, there are some differences. First of all, mitosis ends up with 2 exact copies of diploid cells, meaning they are clones of each other. But meiosis ends up with 4 haploid cells that are different from each other. They are not exact copies. The cells in mitosis copy their DNA, whereas in meiosis, we don't copy the DNA in between, which is why we end up with a haploid cell at the end, or 4 haploid cells, and not diploid cells. If they were to copy, then we'd end up with diploid cells, and what would ultimately happen is we would have too much DNA. Finally, mitosis occurs in one stage of 4 phases, prophase, metaphase, anaphase, and telophase, whereas meiosis occurs in 2 stages of 4 phases each. So we have prophase, metaphase, anaphase, and telophase 1, and then prophase, metaphase, anaphase, and telophase 2. So that's a total of 8 steps. So let's take a look at our objectives and see what we've learned. First of all, what are homologous chromosomes? Homologous chromosomes are copies of chromosomes that are identical to each other, and they're made up of 2 autosomes. The phases of mitosis we just went over. Prophase 1, the cell membrane, or the nuclear membrane dissolves as the chromosomes condense down. In metaphase 1, they line up randomly. We can also have crossing over, remember, in prophase 1, where parts of the DNA switch in place. And then in anaphase 1, the chromosomes split, but they stay intact. They do not separate at the centromere. And then in telophase 1, the cells pinch off into 2. But then we go right into prophase 2 because the cell is not going to allow the DNA to copy, in which case we're going to have the nuclear membrane dissolving, they stay condensed, and then what's going to happen is in metaphase 2, they're going to line up, the chromosomes are going to line up in the center. This time they're going to split at the chromatids during anaphase. Chromatids at the centromere are going to separate, go to each side, and then finally in telophase 2, we end up with 4 haploid cells. The concept of independent assortment is as follows. When we produce our gametes, what chromosome goes into which cell is completely random. That's why in metaphase 1, they line up, and maybe one will go one way and one will go the other, and that's okay, because the final result is a sex cell that's going to ultimately be different than the parent cell. Meiosis and mitosis, comparing and contrasting, all you have to do is look up 2 pages. Here are the similarities between mitosis. They're both forms of cell division. They're roughly the same phases. They both have the purpose of creating genetic material to pass on, and they are asexual processes. The differences being that mitosis ends with 2 diploid cells that are copies of each other, whereas meiosis ends with 4 haploid cells that are different from the parent cell. The cells in mitosis copy their DNA. The ones in meiosis do not. And in mitosis, in one stage consisting of 4 phases, and in meiosis, it's 2 stages consisting of 4 phases each, for a total of 8. And finally, why is meiosis important? Well, if we didn't have meiosis, then sexually reproductive cells would be diploid. And a diploid cell plus a diploid cell is a quadruploid cell. And a quadruploid cell plus a quadruploid cell is an octuploid cell. And an octuploid cell plus an octuploid cell is a sextinoploid cell. I don't do math. Anyway, so I hope you understand meiosis and its importance. This does wrap up our unit on cellular reproduction. And feel free to go back to this podcast or any of the other podcasts in this section to help you grasp the concepts of DNA and RNA, DNA replication, protein synthesis, mitosis, or meiosis. Have a great day.